My name is Bob Brocker. I'm the founder and president of AgeWise Colorado. And Barbara Boyer is on with, she is our executive director. And we're joined today by Dr. Huntington Potter, who's going to be talking to us about Alzheimer's research. So I want to give you a little brief introduction to who we are and what we do. So let me do that real quickly. We're winding up year 4 here pretty soon since we launched AgeWise Colorado. And we're all about connecting people, helping people get connected to great resources, information, education, anything to do related to improving the aging experience of the people of Colorado. And when I say that, I'm including every older adult and every adult child of an older adult. Both have a very, very high level of interest in what we're doing at different times and for different reasons. This is just a quick glimpse at our homepage. You probably have seen this in the past, but if you haven't, please do take a look at our website. I'll give you a little brief bio of Dr. Potter. He's the founder and director of the University of Colorado's Alzheimer's and Cognition Center, and professor of neurology and member of the Linda Crnic Institute for Down syndrome. And prior to coming to CU in 2012, he designed and led a Florida-wide Byrd Alzheimer's Center and Research Institute. He studied, researched, and taught at Harvard University for 30 years and is the author of over 200 scientific articles, books, and patents, and a fellow of the American Association for the Advancement of Science and a founding member of the National Academy of Inventors. His electron micrographs of DNA are on permanent exhibit at the Smithsonian Institute. So if you're in Washington, you go to the Smithsonian, take a look. I'm going to turn this over to Dr. Potter now. He has a slideshow, and we're very happy to have you today, Dr. Potter. And please tell us more about the latest and greatest with Alzheimer's research. Thank you.
Great, Bob. Thanks very much, and thank you all for coming. Now, how come this is not working? Let's see here. Sharing is not turned on.
Is that something I need to do?
Okay, here we go. There we go. Okay.
Okay, got it. You're up.
All righty. So what I want to share with you today are some of the findings that we've had recently at the University of Colorado Alzheimer's and Cognition Center, and in comparison to what are currently available for treatment of Alzheimer's disease that are FDA approved. And as you'll see as we go along, this is really a very large enterprise in collaborative mode with scientists and clinicians, and also working with other scientists and clinicians at various centers around the world. So there's a very important problem to solve, and we solve it in a collaborative way. So the University of Colorado Alzheimer's and Cognition Center has a slogan, which is that healthy brain aging starts here. And that means that we study brain aging, we just study all of the diseases of aging that affect the brain, and we try to develop new treatments and also new diagnostics. So the first report of Alzheimer's disease was made by Alois Alzheimer in 1907 when he studied one of his patients, August Dieter, shown on the left. August had an early form of dementia. She was only in her 50s, but she had the dementia that might be expected of an 85-year-old. She didn't recognize her family, her doctors. And if you read Dr. Alzheimer's report, which is only a few pages long, it's in German, but there are translations. You will recognize immediately the clinical symptoms of what we call Alzheimer's disease now. He didn't name it after himself. The head of his institute did, in order to raise money for studying this new disorder. So in the time since 1907, we've learned a great deal about Alzheimer's disease, and we have insights into what causes it and how it's different from other kinds of dementia. Because right now, because Alzheimer's disease is the most common form of dementia in the elderly, dementia and Alzheimer's disease is often used interchangeably. But in fact, dementia describes the clinical symptoms, and Alzheimer's disease is just one way in which people can become demented. And there are many ways, which we also study at the Alzheimer's and Cognition Center. Now, the problem, of course, is that when Dr. Alzheimer first described his first patient, and for many, 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 many decades afterwards, Alzheimer's was considered an extremely rare disorder because, by definition, it was early onset dementia. But in the 1960s, the clinicians, the geneticists, the biochemists all got together and realized that what had been called dementia of the Alzheimer type, which occurred in people who were elderly, was actually the same thing as Alzheimer's disease. And now these two disorders are combined, and we realize that there are about 6 million people. It's now 7 million in the latest data, patients with Alzheimer's disease or related dementia in the United States, and many, many times that throughout the world. The cost is about $350 billion a year now, and it's expected to go up to about $1 trillion a year by 2050. This is obviously a problem we cannot ignore. It is bankrupting us financially and emotionally. And the reason it's become so prevalent, in addition to the realization that it is all one disorder, is that people are living longer. A lot of advances in medicine have been able to slow the rate of heart disease and cancer. And if 10% of the people over 65 and 35 or 40% of the people over 85 have Alzheimer's disease, you can see why there's been this huge tsunami. Now, women account for about 2/3 of dementia patients and 2/3 of the caregivers. So they're affected disproportionately. And you'll see at the end that we have an idea of why that might be. The other insight that you may not know is that people with Down syndrome all develop Alzheimer's disease pathology in their brain by the time they're 30 or 40, every single one of them. And the majority become demented by the time they're 50 or 60 due to their development of Alzheimer's disease. And we'll see that the reason for that is that people with Down syndrome have 3 copies of chromosome number 21 instead of 2, like everybody else. And the main Alzheimer gene is on chromosome 21, so that's why they're at very high risk. The other thing that you can take away from this slide is that about 60% of the risk of developing Alzheimer's disease is inherited. Now, of course, that means that 40% is due to bad luck, environment, lifestyle, and everything else. But the fact that so much of it is due to genetics gives us insights into how we may be able to understand the disease better and develop new diagnostics and treatments. Okay, so we have at the University of Colorado Alzheimer's and Cognition Center a memory disorders clinic in which people can be assessed as to what kinds of dementia they may have, what other kinds of problems may have. We have 9 behavioral neurologists, 1 even focusing on Down syndrome, and a whole clinical team so that we can see approximately 4,000 patient visits per year. So this is a world-class memory center, and you can see in the map that CU is sitting in the middle of essentially a desert of Alzheimer's disease research centers, where almost all of them are on the East Coast, the West Coast, and then a collection in the middle of the country. So we serve the entire region here in the Mountain West, shall we say. Now, there are drugs that people can take to treat Alzheimer's disease in some ways, but the traditional Alzheimer's drugs really only help nerve cells in the brain communicate with each other a little bit better. I won't go through the details of what these drugs do, but they essentially help nerve cells talk to each other by increasing the sensitivity or the amount of neurotransmitter that they release and which work. But they don't actually affect the disease process itself. And that's where new research over the last 10 or 15 years has been focused on. So here are some of the contributors of our research here at the University of Colorado. You see some of the scientists that are hard at work in the laboratory developing treatments that eventually get into the clinic. And then we see one person who you can tell immediately has Down syndrome. She had a very active life. She was a poet and published a number of books. She had a full-time job, but she developed Alzheimer's disease in her early 50s. And when she died, she gave her brain to science, which we very much appreciate. And it's given us insights thanks to that donation. So what do we know about Alzheimer's disease that allows us to develop new diagnostics, new treatments based on that new understanding? Well, the first thing that Alzheimer saw back in 1907 was that the brain of his patient was much smaller. It had atrophied compared to the brains of people who don't have Alzheimer's disease. And the other thing he was able to identify, thanks to some new techniques that had just been developed, was what we now call amyloid deposits and neurofibrillary tangles. Now, the amyloid deposits are shown here on the top side, where you can see that there's a halo of degenerating nerve cell connections and a amyloid deposit in the middle. And then this is a neurofibrillary tangle, which is inside nerve cells and eventually kills those nerve cells. So that leads to neuronal loss and synapse loss, which underlies, of course, the brain atrophy. And the other thing that Alzheimer noticed in 1907 was that there was inflammation in the brain. Astrocytes, which are not nerve cells, there are other kinds of cells in the brain, and microglia, again, other cells in the brain, they get all activated and angry at this amyloid deposit and try to get rid of it, but they fail in that process. So this is the pathology that occurs in the brains of people with Alzheimer's disease. And sorry about that. They give us insights into how we might try to reverse or prevent the disease. Okay, so here's the brain of an Alzheimer patient, and you can see it is smaller, shrunken compared to a normal brain. And Alzheimer used these new techniques of staining very thin sections of the brain to see the plaques and tangles. And that's essentially what we knew about Alzheimer's disease until about 1984, when George Glenner and Kenny Wong were finally able to identify the major protein that is in the amyloid deposit shown here. And that protein is shown here. These single letters indicate the amino acids that make that protein, and there won't be a test at the end of the talk. You don't have to memorize that. I haven't memorized it. But this is a very major breakthrough, because this A-beta peptide is what causes the amyloid deposits, which then cause the neurons to die and give you Alzheimer's disease. Now, the other thing that came out of this study was very important, which is that this protein is actually derived from a normal human gene. It's not coming from a bacteria. It's not coming from a virus. It's not coming from Mars. It's a normal human protein that is clipped into pieces, and one of those pieces is this A-beta peptide. So that tells us that what is giving us Alzheimer's disease is part of our normal genetic makeup. It just goes wrong in Alzheimer's disease. That protein is very important because using reverse genetics, a number of groups were able to identify the gene that codes for that protein. And that gene, which we call the amyloid precursor protein gene, not a very imaginative title, but it's on chromosome 21. And remember, I told you that people with Down syndrome have 3 copies of chromosome 21 instead of 2. And so that means they have 3 copies of the APP gene, and they start making more A beta peptide. And that's why people with Down syndrome all get Alzheimer's disease. Now, the other thing that this discovery allowed was that there are some families that have inherited Alzheimer's disease. Now, remember, I said that 60% of the risk is due to genetics, but there are some families where if they inherit a mutant gene from one of their parents, they're guaranteed to get Alzheimer's disease. And what was then discovered was that one of those mutant genes is the APP gene itself on chromosome 21. So here's a family, a Swedish family, in which basically 50% of the children of a person with Alzheimer's disease in this family get Alzheimer's disease, males and females together. And that's because they have a mutation in the APP gene, which makes production of the A beta peptide more prevalent. So they have a mutant gene. And that's a tremendous advance in our knowledge of the disease, because it says that it isn't a complex disease, even though it may look that way, but it can be a very simple disease. Extra copy of the APP gene in people with Down syndrome guarantees that they get Alzheimer's disease. A mutation in this family guarantees that if it's inherited in a child, that child will get Alzheimer's disease. Now, there are a number of families like this, and they can get Alzheimer's disease at various ages, depending upon the kind of mutation they have. It can be in their 50s, like August Dieter, or it can be in their 40s or the 30s, or even the late 20s, depending upon the mutation. Now, that's obviously terrible for these families, and we're very sympathetic, but they've made a tremendous contribution to our understanding of Alzheimer's disease, because we can take blood cells from one of those family members, isolate the APP mutant gene, and put it into a mouse. And now that mouse can get Alzheimer's disease. So that's a tremendous advance. So let me just summarize where we are now. The purely genetic, very rare forms of Alzheimer's disease, they're early onset, like August Dieter. Every single person who inherits a mutant gene gets the gene. And we've identified 3 genes in which that can happen. We call them the amyloid precursor protein gene on chromosome 21, or the presenile gene 1 or 2, and they code for enzymes, proteins that actually clip the A beta peptide out of the APP gene. So that's APP protein. And then, of course, Down syndrome, trisomy 21, that's the genetics. And then there are genetics that contribute to even sporadic Alzheimer's disease. That's the majority of Alzheimer's disease. It's just, you happen to get Alzheimer's disease, but it is affected by genes. And one of the most important genes is apolipoprotein E, or E4 in particular. So people who inherit an E4 a gene from one of their parents have about a 3-fold increased risk of developing Alzheimer's. And if they inherit a ApoE4 gene from both of their parents, so now both ApoE genes in their body are ApoE4, then they have about a 15-fold increased risk of Alzheimer's. So this is the most important genetic contributor to Alzheimer's disease. Now, how are we going to find a way to reverse or prevent Alzheimer's disease? We can't just take willy-nilly and inject drugs into people with Alzheimer's disease and see what happens. We need an animal model. And the problem with animal models of Alzheimer's disease is that the only animals that normally get Alzheimer's disease are monkeys that are a little unethical and quite expensive to use for research purposes, and polar bears. And I joke that the dean of the University of Colorado Medical School doesn't want a lot of polar bears running around the medical school. So laboratories across the world were able to take the mutant APP gene from a family that got inherited Alzheimer's disease and insert it into a mouse embryo so that that mouse now grows up with a mutant human gene. It's partially human, one mutant human gene, and the rest of it's mouse. And because of that mutant human gene, that mouse over here on the left gets amyloid deposits as they get older. So that's our model. And in fact, when we put the mouse in this water maze where they have to find this glass platform, which is underneath the water and they can't see it, but if they get out of the water there, they're happy and they can memorize where this platform is if they're a normal mouse, and you can put them anywhere in this water maze and they immediately swim to the platform and get out of the water, unless they have these amyloid deposits and neurons that are sick, and then they can't find that platform. So that's our model. And that allows us to ask questions about diagnosing Alzheimer's disease and treating Alzheimer's disease and maybe preventing it. So using that model and trying to develop new drugs, several companies have developed new drugs that are FDA approved. One of them is called aducanumab, the other one is called lecanemab, and the most recent one is donanemab. Now, these drugs all have the word MAB at the end of them, and that's because they're antibodies. Antibodies are the proteins in your blood that you make to resist COVID, to resist measles, to resist any kind of disease. And you can either get chickenpox when you're young and then you never get chickenpox again, or measles, or you can be vaccinated with a drug that prevents human disease like COVID. So what the scientists did was they made some of these antibodies in mice that were against the amyloid of Alzheimer's disease, and then they changed them into human antibodies, and that's what these drugs do. They're essentially antibodies against amyloid. And they can be injected, and they are very good at getting rid of amyloid in the brain of people with Alzheimer's disease, as we'll see. So these are the newest drugs for Alzheimer's disease. And somehow or other, whoops. Okay, so if you take an Alzheimer's person and you do a PET scan of the brain with a special radioactive molecule that sticks to amyloid, you can see that they got a lot of red amyloid in their brain. And if they get injected with just placebo, meaning salt water, a year later, they still have lots of amyloid, even a bit more amyloid. But if they're injected with any one of these drugs at a certain concentration, 3 mg per kilogram of the person, or 6 or 10, then a year later, almost all the amyloid is gone. So these drugs that attack amyloid are very good at removing amyloid from the brains of people with Alzheimer's disease. So that was a major breakthrough. And the only problem is that they do have some serious side effects. So for instance, they cause brain bleeding and brain swelling, and that's not good. And for some reason or other, okay, this is being a little slow here. Here's the brain swelling and the microhemorrhages. That's not good. So they have side effects, even though they do tend to slow the course of the disease a little bit. So we had another interest, and that was that ApoE, as you remember, is a protein that's running around the body. It happens to carry cholesterol, which is a very important function. And ApoE4 especially is a risk factor for Alzheimer's disease. And what we discovered is that ApoE is a catalyst. And that is to say that it binds to the A beta peptide and teaches it how to make amyloid. So the A beta peptide by itself is pretty harmless, but these amyloid plaques in the mouse are completely prevented if we take away the ApoE gene from this mouse, and that's shown on the right side. It still makes A beta peptide. It just can't polymerize it into amyloid. And this mouse has normal neurons and it has normal cognition. So it has no trouble finding that platform in the radial arm water maze. Now, we can't get rid of ApoE in humans. That's would be very dangerous because ApoE is important. But we had the idea that maybe we could develop a drug that would have the effect of getting rid of ApoE. And here's the experiment that we did. This was headed by Dr. Noah Johnson in the lab. So what he did was he took the A beta peptide, put it in a test tube, and added ApoE4 to the test tube. And when he did that, the A beta peptide polymerized into amyloid fibers in the test tube, so we could reproduce Alzheimer's disease, essentially. And those fibers kill nerve cells. So there's the assay. And we wrote to the National Institutes of Health in Washington and asked them to send us all the known human drugs. And we tried them one at a time to see whether any of them would block this effect. And Dr. Johnson was able to find several that did. And the most successful ones so far are an antidepressant called imipramine and an antipsychotic called olanzapine. And they do prevent AD pathology in nerve cells and in mini brains and in people with Alzheimer's disease that take these drugs for their depression or their psychosis actually get better. Now, we haven't done a clinical trial yet. That will take a lot of money and a lot of effort on many people's part. But at least in the retrospective study, it looks as though these drugs that block the effect of ApoE actually benefit people with Alzheimer's disease. Now, please, please, please do not go to your primary care physician and ask for these drugs for Alzheimer's disease. We have to do a clinical trial to make sure they're safe and effective before they're FDA approvable. But it's promising, and we're pursuing this study. People with Down syndrome all get Alzheimer's disease, and that has given us great insights into how the disease progresses and also has helped develop those drugs against the amyloid deposits. But we were also interested in finding people who maybe were protected against Alzheimer's disease. And it turns out that people with rheumatoid arthritis tend not to get Alzheimer's disease. The question is why? It was hypothesized that because they take lots of anti-inflammatory drugs, and we knew from our work and others that inflammation was part of Alzheimer's disease. Even Alzheimer himself noticed that. Maybe by accident, they're preventing Alzheimer's disease. So a very large clinical trial was started, funded by the NIH, to use these kinds of drugs in people with Alzheimer's to see if it helped. And the trial totally failed. So the idea was reasonable, but it didn't work. So what we did was we started looking in the literature for anything about rheumatoid arthritis that might give us a hint about why they were protected from developing Alzheimer's disease. To make a long story short, we discovered a protein, which is a natural human protein, called GM-CSF, granulocyte macrophage colony stimulating factor. It teaches the bone marrow to make more granulocytes and macrophages. That's its normal function. And it turns out that granulocytes and macrophages, when they get activated by GM-CSF, are very good at getting rid of amyloid in the brains of our mice. So here's a mouse brain, and GM-CSF was injected in this side and some control solution on this side. And 1 week later, almost half the amyloid was gone. So that was a breakthrough. And the exciting thing was that GM-CSF could be injected subcutaneously. It crossed into the brain from the blood, and we could ask what happens to the cognition of these mice. And it turns out that in this radial arm water maze, we asked how many errors the mice made, and if they contained that mutant human gene, so they had amyloid deposit, they made lots of errors. But if they were treated with GM-CSF, they made the same errors or even fewer errors than a normal mouse, shown in green. And even the normal mice were benefited by this drug. So here we have a drug that gets rid of amyloid and makes the animals have normal cognition. It's a known human protein. It's also a known human drug because since it stimulates the bone marrow to make granulocytes and macrophages, that's a useful thing to do. And several companies have made human GM-CSF as a treatment for bone marrow problems. So we could start a clinical trial with an FDA-approved drug for GM-CSF. And here's the results of our first trial, published a couple of years ago. People were chosen randomly who had Alzheimer's disease. Some of them were given GM-CSF. Some of them were given saltwater injections. And 3 weeks of treatment improved the memory of the people with Alzheimer's disease if they took GM-CSF by several points. And even 45 days after the end of treatment, they were still better than the placebo group. So here's a treatment that actually improves cognition in people with Alzheimer's disease. The other exciting thing is that we could take blood from these people and we could ask what happened. And it turned out that the amyloid in the blood increased when they were treated with GM-CSF because it's being taken out of the brain. Tau, which is a protein in the neurofibrillary tangles, that actually went down with treatment with GM-CSF because the neurons are not as sick as they used to be. And here's a protein called UCHL1, which is released when neurons die. And people with Alzheimer's disease have more of this in their blood. And when they're treated with GM-CSF, the amount goes down drastically. So they're essentially stopped from killing their nerve cells in the brain. We also asked the question whether GM-CSF might improve cognition in normal aged people by studying wild-type mice that are aged. And it turns out that GM-CSF improves cognition in Down syndrome mice and in normal mice that are just old. So it seems to be a general cognition enhancer. And that's also exciting that maybe we should try it on everybody over 70. So that's a trial that's being planned now. So how does it work? How does it actually help the mice with Down syndrome, the mice with Alzheimer's disease? Well, it turns out that if you stain for nerve cells in, let's say, a Down syndrome mouse, you can stain nerve cells in the cerebellum, for instance, or in the hippocampus, and they stain very well with certain stains. But in the DP16 mice, these nerve cells are gone. And that's after treatment with saline, but it would be before the treatment with saline. They just don't have these nerve cells. But if they are treated with GM-CSF, pop, 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 all the nerve cells come back. Now we look carefully and we think that the nerve cells are there here. It's just that they're not active. They're not functioning properly. And when the GM-CSF is used to treat them, now they're functioning properly. And that's shown on these slides here, where the number of these nerve cells in the cerebellum are perfectly normal in a wild-type mouse. In a DP16 mouse, they're much fewer. And then when GM-CSF is used to treat the mouse, then they all come back. Well, not all, but a lot of them come back. The same thing happens in the rat model of Alzheimer's disease. I won't go through these figures in great detail, but here, the wild-type mice have very few neurons dying. The Alzheimer's rats have a lot of nerve cells dying. And when they're treated with GM-CSF, the number of nerve cells dying goes way down, almost to the level of the wild-type mouse. So we basically turned a rat model of Alzheimer's disease into a normal rat again. Now, what happens in people? I said that people with Alzheimer's disease had a lot of dying nerve cells, and the GM-CSF treatment in the clinical trial stopped that. So what we did was we asked, what happens over your entire life? How many nerve cells are dying just normally as you live? And it turns out that nerve cells, especially in women in the red bar, are continually dying from age 2 to age 85. In men as well, they're continuing to die. It just gets faster and faster. Here's another measure of neuronal damage, the neurofilament light protein, and it again rises exponentially with age. So your brain is always dying and causing problems for its nerve cells. And here's a measure of the inflammation. The inflammation in the brain begins to rise, especially in females, after about the age of 30 or 40, and males also rise. So we have three measures of neuronal age or brain age: neuronal death, neuronal damage, and inflammation that all rise exponentially with age. So what happens in those people with Alzheimer's disease when they were treated with GM-CSF? They got better and there was less nerve cell death. How much? Well, it turns out here are the Alzheimer's people before they got treated, and here are the people after they were treated with GM-CSF compared to the normal progress of brain aging. And you can see that we've essentially turned our Alzheimer's people into 5-year-olds in terms of neuronal death. So it's completely halted the neuronal death in the brains of these Alzheimer's patients. So that's very exciting, and we will see what our next clinical trial is telling us. The first trial was only for 3 weeks of treatment. This one is for 6 months of treatment in people with Alzheimer's disease. It was funded by the Alzheimer's Association and the NIH. And they get 7 injections a week for 24 weeks. It's been approved by the FDA, approved by the Institutional Review Board, the University of Colorado. And we started screening in 22, and we think that the entire trial will be finished in 2026. We've got 15 participants so far completed. We need about 40 total. And if you're interested or a friend is interested in participating in this trial, you can take this 2D code and put it on your cell phone and get hold of us. Now, we've been talking about the genetic risk of Alzheimer's disease, the potential treatments for Alzheimer's disease like GM-CSF or imipramine. But what can you do while you're waiting for our trials to get finished? Well, remember that I said that 60% of Alzheimer's disease risk is due to genetics. But there are a lot of things that increase or decrease your risk that you have better control over than your genes. And here are the list of things that give an increased risk: high blood pressure, stroke, high cholesterol, smoking increases the risk about 2-fold, diabetes increases the risk about 2-fold, low educational attainment, go back to school or come to meetings like this, high BMI, body mass index in midlife. All these things are increasing your risk. You can decrease your risk by exercise. And we've found that caffeine is helpful in the mice and possibly in the people. And of course, keeping your brain active is good. So here's the study that we did with the environment. Here's a typical mouse cage. They get food and water. They get Obamacare. They're probably treated better than most humans on the planet, but it's a little lonely, a little boring. But if we put the mouse in the Club Med cage like this, where they have toys, which we change every week, and other mice to play with, running wheels, then they get better. Then they get better. And that's very exciting because we can change our environment by participating in social activities, getting more exercise, stimulating the brain. Here's what happens when we treat mice with caffeine. It turns out that it reduces the amount of A beta in cell culture and in the mice. That's shown here. And here's the amount of A beta in the brains of the mice. And if we treat them with caffeine, the amount of A beta goes down and the mice get better cognitively. So we're not really recommending people go off and start drinking 3 to 5 cups of coffee a day, but at least in the mice, it definitely helps. I want to thank everybody who's come to this webinar and also all the people who participated in the research that you've heard about. Um, it's a real team effort with scientists and clinicians, and especially research participants and their families, because without them, we couldn't carry out any of this research. And I must also thank the philanthropists and the funding agencies. We first published our GM-CSF mouse study in 2010. And we finally got an NIH grant and an Alzheimer's Association grant to study that in about 2022. All of the work that was done between 2010 and 2022 was done by research funded by philanthropists. Because we went to the NIH, said, we've got this mouse and it looks like it's benefited by GM-CSF. Give us some money for a clinical trial. They said, oh, tut, we have a lot of ideas and go back to your mice. And so we had to do it ourselves. And that's been very successful. We really appreciate the help we've gotten. So the hope is that with a few more years of work, we will be able to announce that the University of Colorado has, if not a cure, at least a treatment for Alzheimer's disease that reduces the amyloid, reduces the neuronal death, and improves cognition right here on our campus. So thank you very much for participating. And I'm going to stop sharing here so that you can actually see me. And anyone who wants to ask questions, or if you wake up at 3 in the morning and you want to ask a question, go ahead and email me. I may be awake and I can answer, or answer in the morning. You should consider the Alzheimer's and Cognition Center your source of information. And if you read on the internet that broccoli cures Alzheimer's disease, don't believe it and get hold of me.
Thank you very much, Dr. Potter. This is fascinating. And thanks so much for all the work that you and your team have done to bring it along this far. We have a question from Kelly, and she asks the status of the leukoin study and its potential for approval for treatment.
Yeah. Kelly, thanks for asking. Leukoin is the commercial name of human GM-CSF. So when I told you about our current trial, it is ongoing and we're still recruiting people. We hope to finish sometime in early 2026. I don't have any data to share because, of course, it's a double-blind, placebo-controlled, randomized trial. I don't know who gets the drug. They don't know who gets the drug. And so we can't give you a sneak peek that would destroy the study. So we'll have to wait for me to be invited back by Bob for another Age-Wise Colorado talk in about 6 months or a year. Call it a year.
A year sounds good. Yeah, well, I'd be happy to have you back. And so, one question that popped into my mind when you were talking about the GM, GSF, leukoin is this something that people might want to take just to slow the progression of nerve cell loss, period, without having Alzheimer's?
Yes, that would be our next trial. We need to raise about $3 or $4 million to try to do a trial in normal aged people who already have nerve cells dying in the brain. And we would predict that GM-CSF treatment would stop that neuronal death. Now, the question is, do you want to take it for the rest of your life, or can you take it for a few weeks and then take 6 months off? All of those kinds of clinical trial research questions will take some more effort. So please, if you walk down Colfax and somebody says that they're willing to sell you some GM-CSF, don't believe them. Please don't take it. We got to finish our research first.
So once you finish your research, um, and then, you mentioned earlier, you discovered this in 2010 and you got funding for a clinical trial, essentially 12 years later. So what's the time period between finishing your research and saying, let's say it turns out that, oh, yeah, this is the greatest thing ever, and so we need to proceed. Then, how long does it take to actually get it out to the public and start, if people can start using it?
Well, it's all a matter of collaboration with pharmaceutical companies who would have the resources to do a phase 3 trial, which might cost several $100 million. And we're talking to them and they're talking to us. So they want to see the second phase 2 trial finished. And then after that, it might only take a year or two to get everything done if you throw enough resources at it. Um, if this were a head-to-head trial of the monoclonal antibodies, the anti-amyloid antibodies versus GM-CSF for safety and efficacy, we might win, and we'll have to see when this trial is finished.
So I don't want to put you on the spot, but the most, well, kind of will. So the most, optimistic outlook in terms of what year, if everything falls into place, the phase 2 goes well, drug company picks it up, they do the phase 3, it all works out. What year are we looking at?
4 or 5 years from now.
Okay. Well, that's not very long for some of us, and it's a long time for others.
Right. And the thing about Alzheimer's, of course, is that it develops probably 20 years before the first clinical symptoms. So, many people who are in their 60s and 70s have Alzheimer's and just don't know it. And if this drug or any of the others that we and other scientists are working on can also prevent Alzheimer's disease from developing, that addresses the question that you asked is, should people start taking drugs to prevent Alzheimer's disease in their 40s or 50s, so as to never have a risk? And that remains to be seen. That would be the hope, but it remains to be seen.
I asked a question in some other forum once about, why does the human body only last 100, or so years, maybe a little longer with some people, but, and it was, the answer was, well, it's cell death, well, you mentioned the nerve cells, but, so if you prevent nerve cell death, you could live a lot longer, I would assume, right?
Yes, because we do know that nerve cells can be replaced. It doesn't happen very effectively, but it does happen. So if you prevent them from dying and you allow them to continue to rejuvenate or regenerate, then yes, your brain may last. Whether the rest of your body lasts is another question. But we know much more about how other cells in the body have stem cells that can replace them. So there's a lot of research on the question of what is real aging, why does it happen? Of course, from a geneticist point of view, there's no selective advantage to living to be 100. You've already had your children, they're gone, they're having their own children. So the only thing that evolution wants is for you to live to be 40 and then stop being a burden on your children so that they can have their own children. So living to be 100 is not selected evolutionarily, but we're scientists and we can try to intervene to help people live longer and healthier lives.
Yeah, that's the key. I mean, I saw another chart recently that showed the difference between lifespan and health span. It was like 10 years. But
Yeah, for some people it's zero. They continue until they fall off the twig and they're healthy one day, and then they pass the second day. But most of us go through a long period of lesser activity, shall we say.
Mhm. Well, I like what you said earlier too about, what things you can do to kind of either delay this um, or prevent it in some cases. Pretty simple stuff, get out there in the world, exercise and eat better. And uh, I never thought about the caffeine thing, but that's interesting. So you may make Starbucks a lot more money eventually.
Well, we talked to them about it, and but they didn't want to support the research because they didn't want to be on record as saying that caffeine is a drug. They want
Mm,
to think of it as a lifestyle.
mm.
And so, yeah, they passed.
Well, they could market it as a lifestyle enhancement, whatever, but yeah. Okay. Do we have any other questions from anybody on the call before we sign off?
Seem to be four chats. I don't know what they're saying. Oh, thank you. Okay, well, that's good. Share the recording. Yes, I talked to Bob. He guess he has a recording. It's okay with me.
Yes. Yeah, we definitely will. The recording will be on our website in a few days. So yes, and you'll be able to access it there for everyone, including people who are not on this call. So yes. Okay, any other questions or comments? Okay. Well, hearing none, once again, thank you very much, and we'll be doing this again next year. So, and
All
getting
right.
the latest updates. All right, we'll
Well,
keep up
later.
the
Thanks for
work, Dr.
coming.
Potter. Thank you very much. All right, have a good day. Take care. Bye bye.